All right, hi Paul. Actually, hi everyone. Welcome back to CVRG. This week we have, I guess, a special guest, Pol Castellano. And actually, Paul's not really a guest because he is a key member of the CVRG team and has helped write all the code for the previous sessions. And sorry, I'm just saying. So today, Paul will introduce or describe a package that he has made called POMA that allows for the easy pre-processing, handling, and statistical analysis of metabolomics and proteomics data. So Paul, before you begin, I have a quick question. Why POMA? What does that mean?
That's I think is a difficult question. It should be an acronym, but actually, is the name of is Apple in Spanish? Actually, in Catalan. So
Oh,
yeah, I wanted that to be an acronym of different omic things and some biology stuff, but at the end, so for now, it's just POMA. Is like the say is Apple in Catalan. It's,
oh, nice. So is there? I haven't actually. I don't remember what the hex sticker looks like. Is there an apple on it?
yeah, is it?
Okay, okay, that's why. Yeah, Poma, Pomodoro, Pom. Okay.
Yeah, yeah,
Oh,
kind of the same exactly.
nice. Oh wait, no, Pomodoro is tomato. Anyway, I don't know what I'm
Actually,
talking about.
actually never related with omics or something like that. Just an apple.
Nice. All right, Paul. So I guess I'm going to share your screen so that you can go through your presentation.
So yeah, can you see my screen?
Um, yes.
Okay. Okay. Should I start?
Yes, please go ahead.
Thank you. Yeah, thanks actually for the introduction. My name is Paul. I'm a postdoctoral researcher at Duke at the Duke Molecular Physiology Institute, working in Hershey Lab with Akshay. And yeah, today I'd like to present this software, this called POMA, that is a user-friendly workflow, both using one bioconductor package and one Shiny application for the omics data analysis, basically. So say that POMA focus on the statistical analysis of this omics data, not on the first operation of the omic workflow, meaning, for example, peak integration, and none of the last operation of the workflow that can be, for example, enrichment analysis. So POMA is just focused on the middle part on this statistical analysis. So we have different ways in R to approach our statistical analysis. So we can work directly, for example, using our Excel files, CSV files, etc. This is the worst way because we can introduce a lot of error during this Excel file manipulation or all of this. So we have another way that is so we can have our data stored in bioconductor objects like MSNZ, Q features, summarized experiment. So actually introduce all of these objects before in the CBRG meetups. And then so if we have our data in this object, we can extract the data from this object and then adapt or format our data to be used for other R packages to perform the statistical analysis or other functions or whatever. But so this is better than the first one, but it's not optimal because so we can also introduce the error in our data during this manipulation process. And finally, we can use directly these bioconductor objects for our analysis. So we can use as an input these bioconductor objects for visualization, for the statistical analysis, for everything. Yeah, this is the most reproducible way and most interoperable way between different bioconductor packages. Say that the input required in POMA is a summarized experiment. So actually, as I said, he introduced very well this type of objects in a previous meetup, CVRG meetup. So probably you will know, but for this summarized experiment, for this POMA input, we need in this summarized experiment basically two files: one target file that is the summarized experiment call data, and one features file that is our assay. In our call data, we need the sample IDs in the first column, our group, that is a factor variable. So all the groups in our data. And from the third column, we can include different covariates. This is optional, but we can do that. So we can include different covariates in our call data. On the other hand, our feature file or assay file is only for our variables, let's say metabolites, proteins, genes, whatever. In this case, we will be working in this for this presentation in this metabolomic is a sorry metabolomics data set for colorectal cancer. So we have all of these metabolites and three different groups in this analysis. So if we don't have a summarized experiment, POMA offers us this function to create this summarized experiment using two data frames, one for the target file and the other one for the feature file. So let's create this summarized experiment named my summarized experiment and start the analysis. First of all, when we have this summarized experiment, we need to pre-process before the statistical analysis. We have to pre-process our data. What's first? First, we have to deal with our missing values. This is super common in omics, but probably more in metabolomics, proteomics. So often we have a lot of missing values, so we have to deal with that first. POMA offers us this function, this simple function that allows us to deal with these missing values, to treat zeros as NAs as real zeros, remove features with a lot of missing values under a certain cutoff. For example, here we are considering our zeros as real zeros and not as NAs, and removing those features with more than 20% of missing values. And with for those remaining missing values, we can use 6 different methods to impute these values. So we can do that all of these using just this function. The next step of this preprocessing is the normalization. So again, the input for every function in POMA is a summarized experiment. So using this single function, this easy function, we can normalize our data using, again, 6 different methods for normalization. So using only these two functions, we can go from our messy data with missing values and non-normalized to a summarized experiment that is without missing values and normalized. But there's one step missing in this preprocessing, that is the outlier detection and cleaning. A lot of people don't like this step because it's super hard to obtain the data. And when you told, we have to remove some samples from the data, it's not, no, please, we cannot do that. Because I understand that sometimes, depending on our purpose, maybe we should do that. It's not mandatory, but sometimes depending on our purpose, we should consider to remove some samples from our data. So again, the input is a summarized experiment. This summarized experiment, preprocessed summarized experiment, and using this POMA outlier function, we can do two different things with the same function. If we set this parameter to analyze, the output of this POMA outliers function will be like different tables and different plots to explore our outliers, all the outliers in our data for each group. But otherwise, if we set this do parameter to clean, the output of this function will not be tables, plots, and anything of that. So the output here will be a summarized experiment, a clean summarized experiment without sample outlier. Okay, as I said at the beginning, we are working here with a metabolomics dataset for colorectal cancer with 3 different groups: colorectal cancer, healthy, and polyp. But we may be interested in only 2 of these 2 groups. Maybe, let's say, so we can see here we are able to identify some outliers for each of the 3 groups. But maybe, as I said, we are interested only in colorectal cancer and healthy. Luckily for us, this is a summarized experiment object, so we can do the following. So this is the number, this is our summarized experiment, our preprocessed object, and this is the number of samples. So running this line of code, we can subset everything related, all the data related with colorectal cancer group and healthy group. And this is the number of samples now. So the great thing of working with this summarized experiment object, because doing that with just one line of code, we are excluding all the information related with the polyp group, and then we are keeping the information related with the cancer and healthy group. So now what we have is another summarized experiment, a subset of the first summarized experiment, only containing these two groups without missing values, normalized, and without outliers. So, okay, for now the preprocessing is done. So let's move to the not statistics yet. Let's explore a little bit our data before starting with the statistical part. Sorry. For example, for sample exploration, we can do this. This is this clean object. I call that clean object. So this is our clean summarized experiment. Still a summarized experiment object, but a clean one, the preprocessed one. So we can do this. For example, we can use this POMA boxplot function, grouping by samples, and we can do the same for using POMA density function, grouping by samples. And the output here is all of our samples. These are all of our samples for the two groups of interest, our subset in this case. So we can see that the effect of our normalization, it went really well, because we can see all the samples centered here near zero. And we can do the same using for the distribution. So we can see the distribution of our two groups of interest using this POMA density function. This for samples, but maybe we are interested in features in our data. So again, using the same function, the function is exactly the same, the input is exactly the same, but we are just changing this group parameter to feature instead of samples. And what we have here, so we have again our two groups of interest, but instead of samples, we have here all of our features, all of our metabolites in the data. And here, using this POMA density function again, but grouping by feature, we have the distribution after normalization of all of our features in the data. But often in omics experiments, so this is really common. So we have a lot of features in the data, and basically here we can see nothing, and maybe we are interested only in some features in our data. So again, using the same function, we can do that. This is again the same input, the same function, the same group parameter, because we want to see to display our features, and we can do that. We can say, okay, feature name, plot only these three feature. Actually, this is a vector, so we can create this vector outside of this function, but we can do that also on the fly. So if we set this parameter, we put some vector in here with naming some of our features. The output of this function will be only the box plots for our features of interest. What else? This is regarding heat maps. So again, we are using all time in all POMA functions. The input is the same, a summarized experiment, the preprocessed one, and we can plot a heat map super easily using this function. Actually, behind this function is running complex heat map package. That is a bioconductor package. Is a great package to plot heat maps. So we are using behind this package. But as you can see, we are not using any parameter here. We are using all the default parameters, and only running this, we can plot this heat map here, and you can see our group. This is our most important variable, because this is, as we can see here, this is our two groups in our data. So we can plot this heat map. But if you remember, we had different covariates, different other variables in our call data. So we are working here now. This heat map is clustering our samples using this group here. But we had different, and this group here is this one. But we had other covariates, for example, age, that is numeric, but we have gender, smoking, and alcohol that are factorial variables. So we can include that if we want in our heat map doing this. You see, this is the same function, same input, but I'm telling this function, okay, calls. This is call 1, 3, 4, 5. I'm telling this function, include the column 1 and 3 to 5. And here we can see the same heat map, but including the clustering or this strip for any of our variables, factorial variables of interest. Actually, maybe this is not a great example, because as you can see, it's very difficult to cluster our samples um, within clear groups. So we don't have a clear separation in here, maybe here, but anyway, maybe it's not the best example, but it works anyway. And finally, to close this exploratory analysis part, I'd like to include here a volcano plot. I know that we have to do some statistics to plot a volcano plot. Actually, here we are performing a t-test in order to obtain the data to plot this volcano plot. But still, we can use volcano plots for exploration, because using let's say easy statistics, we can display at the beginning of the analysis our potential most important features. So for example, here we can see three metabolites that are down regulated and one metabolite that is up regulated. So for this comparison, so we can say it's not our final statistics, our final results, but we have an idea of some of more our more interested features. And finally, statistics. That is the strong part in this software, the strong part in POMA. I will not talk about all of this. So these are all methods provided for now in POMA. We have different univariate analysis methods, multivariate analysis, cluster analysis. For now, only k-means, but we are working in more methods for clustering. We can feed lemma models using POMA. So actually, again, introduce this lemma methodology before in these meetups. So we can do introduce the way of doing that using lemma, the original lemma model. So we can do the same here, models with and without covariates, but in a super easy way. We have different correlation analysis methods too, regularization methods as lasso, ridge, and elastic net, allowing external cross validation. This is important. The same for random forest. We can fit like classical random forest classification algorithm, allowing this external cross validation. I'll talk about that in a couple of slides. Finally, rank products and odds ratio for binary outcomes. So for our two group analysis, if you have two groups, we can fit this odds ratio model. This is more a concept than a feature or a function. So all POMA functions are focused on simplifying and compacting the analysis. And what does that mean? So, for example, here for POMA, oops, sorry. Um, yeah, okay. For example, here for POMA univariate, we have only one function for all the univariate tests in POMA. Okay, because it's not we can fit, so we can perform all the univariate methods offered in POMA using the same function and only changing this parameter. Why? Because this is, I think this is useful for two things. First one is for the developer of the package. So it's easy to maintain the one function than to maintain four functions. So from a programming or software point of view, I think this is much better. And also for the user, because it helps user to understand what he or she is doing at every point of the analysis. So using this function is helping us. For example, we are performing an univariate test. Which kind of univariate test? It depends, but we know that we are performing an univariate test. And the same, for example, in POMA multivariate, we have the same function, and using the same function all time we can perform a PCA, PLSDA, SPLSDA, etc. So as I said, this is more a conceptual thing than a feature, but I think it's interesting to say that. And regarding this external cross-validation, POM allows us to split our data within train and test set if our purpose, the purpose of our analysis is a predictive purpose. For example, we can fit a lasso model, setting this n-test parameter to null. If we do so, here we are fitting a lasso model using all the samples in our data. So it can be useful, for example, for feature selection purposes, or so it can be great. We are using the whole data for this model. But what happens if our purpose is predictive, is to build a prediction model. So we need a real prediction matrix without overfitting. And we can do that. Using the same function, we can set this n test parameter to 20. And here we are splitting at the beginning of the function, we are splitting 20% of our data, keeping this 20% of our data, then building a model with the other 80% of the data, and then using this 20% of the data only for the validation. This way, we are obtaining real prediction metrics without overfitting. And we can do the same, for example, in random forest, in this classifier. We can set this n parameter to 20. So the model is built using the 80%, and the validation, the external cross validation, is using this 20% of the data. And these features, these are the number of samples. So this is our 20%. You can see here our confusion matrix with the error in each group, but this error is real. I mean, this is non-overfitted error, because we never use this data to build the model. Again, this is just a reminder. These are our call data, our target file, a subset. So we have only two groups here. This variable here, these are our samples, and we are working here so far. For example, here, these models, our outcome here in these models is this group variable, this cancer and healthy groups. But often we have different covariates in our omics studies, and often we forget to include these covariates in our analysis, or we don't want to include these covariates, or we don't know how to do that, or etc. So and these covariates can be really, really important in our analysis, I would say, especially in metabolomics. So it can be useful to include some of these covariates in the analysis. But I know that doing that, coding that in R can be sometimes tricky or difficult, but we can do that in a very easy way. For example, using this software. For example, here, this is again, I know I'm repeating this a lot of times, but I think it's important. Here, the input is the same input. It's a summarized experiment object. This function is I've seen this function before, this POMA univariate function, and the method is ANOVA. So far, till here, we are fitting an ANOVA model. But what happens if you do this? If you set this covariates parameter to true, and then we put here the covariates we want to include. Then with these two lines of here, with these two parameters, we are fitting an ANCOVA model instead of ANOVA model, because the default, if we set these covariates parameters to true, we are including here all of our covariates. So age, gender, smoking, alcohol, all of these. But if we set this parameter diff, if you select our covariates of interest here in this parameter here, we can include only this subset of variables. So with this line of code, only using this function, we are fitting an ANCOVA model. Feature is, in this case, is a metabolite. So we are fitting this model here, age, gender, and our group, that is our most interesting variable. And it's pretty easy to do that here. So we can do the same, for example, in Lima. So we can fit a Lima model for this contrast, healthy against colorectal cancer, including covariates, and including only, now instead of gender, we are including smoking, like age and smoking. Doing that with this line of code, we are fitting this model here, this Lima model here. And we can do the same with odds ratios. This is slightly different because the outcome here is not our feature, is our group. So we need a binary outcome. We need this two group cancer and healthy outcome. So if you do that, we can fit, if you have a binary outcome, you can do that. You can use this function, POMA odds ratio, and then set this covariates to true again, and then indicate here now three covariates: smoking, alcohol, and age. So we are fitting this model now. Our group, that is cancer or healthy, smoking, alcohol, age, and all of our metabolites. So we can indicate here also which metabolites to include. The default is include everything, but we can do that. We can do the same here for call data variables. We can do the same in this function for our metabolites or our features. And this finally, this is a use case. This is a real use case, and how to do the whole analysis using POMA. So what we have here, this is a summarized experiment again, but containing all the raw data. So this is a messy object full of missing values, non-normalized, with maybe some sample outliers, etc. Using this as an input, this summarized experiment, we can deal with missing values with this line of here. We are imputing using the KNN algorithm to fill all of these missing values in our data. Then we can use this second line to normalize our data. We are performing a log scaling here. And then we can clean the outliers of our data using this third line of code. And finally, we can feed the output here at this point is a preprocessed summarized experiment. So now we can run this line of code and then adjust a lemma model for this comparison, healthy versus colorectal cancer, and using all the covariates, because the default, I said the default, when you set covariates to true, the default is include all covariates I have. So on fit a lemma model, and um, these are our statistical results. So you can see our p-values, log-fold change, etc. So we can go from a summarized experiment object with missing values, non normalized for everything, our messy object, to our statistical results, doing like running only these 4 lines of code. And I think it's pretty easy and pretty readable. And finally, I would like to introduce this software. So POMA is as I said at the beginning, POMA is a bioconductor package. So we can do the whole analysis in R. But what happens if we don't know how to code, we don't want to code, or we prefer to do our analysis using an interactive application? So we have this application that's called POMA Shiny, and this is the interactive version of POMA. So in this application, so all POMA functions, everything in POMA is implemented in this application. So we can do exactly the same using R or using this interactive application. So let's take a quick look in here. This is the home page. Then for uploading the data, we can work in POMA Shiny. We can use different example data sets, or we can upload our own data. You can see here our target file, our feature file. Actually, we can subset here. Let me. Okay, so actually we can subset if we upload the target file, for example, with 4 groups, and we are interested only in 2 groups, we can select the rows like associated with only these 2 groups, and then subset the data in here on the fly. So we can do all the same functionalities in the POMA package. We can do all the same using POMA Shiny. Uh, okay, once we have our data, we can go to the preprocessing step. As you can see, we have missing value imputation, normalization, and outlier detection, the 3 steps in here. Sorry. Okay, yeah, this is our data, imputed data. These are our methods for normalization. This is our normalized data. We can go here and see the effect of our normalization in this box plot. We can see the effect of normalization. It looks pretty well. And all plots in POMA are interactive plots, so we can zoom in, zoom out, download plots, do everything. We don't need to go to outlier section, because by default, this software is removing all the outliers in our data. Is the default, so we don't have to do nothing here. And then we can go, for example, to multivariate analysis, and yeah, let's do a simple PCA here. Sorry, here we didn't, if you remember, we subset. Okay, we didn't subset our groups here, so we still have this polyp group, and then for this reason you can see 3 groups on here. And finally, we can fit a random forest here using this 20% of the data. So you can see all plots and all tables here, our results for this random forest classifier. But we are using here this 20% of the data only for the external cross-validation. So all these metrics, prediction metrics, again, are real prediction metrics without overfitting. And finally, This, if you're familiar with R, this is like a help panel in R. So for each of these function in each tab of POMA Shiny, we have this little helper that, yeah, the aim of is like guide users a little bit through the analysis. So you can see these helpers in each step of the of this software. Yeah, you have here the link, the GitHub link of all the code, and everything is open source, so you can do whatever you want with this software. So, like to finish, POMA provides us a robust, reproducible, and user-friendly workflow for omics data analysis. POMA allows the easy use of covariates in the analysis, so can include covariates in the analysis and adjust for these covariates in a very easy way. The POMA Shiny is the interactive Shiny application of POMA. Everything we can do in POMA, we can do that in POMA Shiny as well. And finally, POMA and POMA Shiny are both open source tools, so everybody is welcome to download, contribute, wherever. Yeah. So thank you for listening, and I'll be happy to answer some questions or whatever.
That's great, Paul. Very clear and thorough presentation. I think the most impressive thing for me was the slide where you showed how you can just chain together, I think it was what, 4 lines of code to go from your input data to pre-processing, cleaning, normalizing, and then you do some statistical analysis. I think that's amazing. I
Yeah.
also think this is a really great opportunity, I think, in future to have some sessions dedicated to describing like the advanced machine learning concepts you went over, like random forests, multiple regression, things like that. So yeah, thank you so much. I think now, so what we're going to do now is like I'm a novice POMA user, so I just kind of want to show you how to run this in our studio. And because I'm technologically challenged, Paul can't actually see my screen right now. So hopefully I do a good enough job of describing what I am, and then Paul can correct me if I say anything wrong. So the code that I'll be showing has been this code has been copy pasted from one of a few really helpful vignettes that Paul has written. So Paul, this is the POMA demo vignette. Could
Okay.
you describe the data set that's used there? So I'll be using the ST something something data set.
Yeah, both are called ST something
Okay.
something.
Oh no, this is
There's
the DMD one.
sorry. Go ahead. Sorry.
This is the DMD one.
Okay, okay. So I use here, that's perfect because I use here for this presentation the other one that is a colorectal cancer metabolomics dataset with 3 groups, and yours is another metabolomics dataset with only with 2 groups, more missing values, kind of different. Yeah, less samples. So it will be great.
Perfect. So it's DMD and healthy samples, no additional covariates.
No,
Okay, so simple.
there's one. I think that there's one covered.
Okay,
If I'm
we'll find out.
yeah.
All right, so I'm
Should
gonna.
I stop sharing now? Sorry, actually, should I just stop sharing or my screen?
I think
No.
it should be fine.
Great.
Okay, so hopefully people can see my studio, and if you can, as usual, we're going to move to just use. Sorry, we're going to open up our CBRG project or the project that's associated with these meetups. And if you don't have one, just open up our studio. So as you can see, I'm in the CBRG meetups project folder. So I'm just going to clear this. And as usual, let's reinstall or if this is your first time, install the CBRG package. So that's remote colon colon install underscore GitHub, and then it's Duke CCT slash CBRG. CBRG is in all caps. And then just specify that force equals true, because in case you have this package installed, you want to overwrite it. So just run all of this in your console. We don't need to open up a new file for this. All right, so all looks good. And then we just run library cbrg to launch the package. And within the package, there are some functions. The one we'll use is called launch underscore project, and this is project number 12. And if all of that works, you should see an R Markdown file that looks like this. Before you begin, make sure that you save as, and then save this in your project directory. So CBRG meetups. And the reason we do this is if you don't do save as and you just save this our markdown file, it'll get saved somewhere else. So this way, it's saved within our project folder. And then again, once again, this is all really nice code written by Pol to install and load the relevant packages for today's session. Once again, keep an eye on your console because when R is installing things, it'll very likely ask you some questions. In my case, I already have all of these packages installed, so it just went through the process of loading them. So as I mentioned, this code and text have been taken from a vignette that Pol has written for this package. Now, there's one function, really useful function called browse vignettes. So we do browse vignettes, and then within inverted commas, you give the name of the package you are using. So if you do browse vignettes POMA, I'm not going to run this because this will open up a window in your browser, and then you will see all the vignettes that are associated with this package. Now, some packages don't have vignettes associated with them, so either you'll get an error message or you won't see anything. In this case, if you run this, you'll see that Pol has written 3 vignettes. And you can either view the vignette in code form, like this, or you can view it in HTML form, so it's nicely formatted. So this is a really useful function, and just as POMA has a few useful vignettes, so do other packages like dplyr or ggplot2. And then, so this is a really useful way. It's an alternative way to just Googling, ggplot2 vignette. So that's a useful function to know of. And so here you can see that Pol has nicely described the workflow. I'm going to skip this part. And he shows you how you can either use your, if you happen to have CSV files that have your data, you can read them in like this, and you can make the object that you need to make to run POMA on. Or you can use I believe it's an MSN set object. So if we go to line 91, that's what we'll do. So if we just do data, and the name of this object is ST00336, and we can see that when we run it, it appears in our environment pane.
It used sorry, it
Oh,
used to be an SM set one.
okay,
Now it's a summarized experiment one.
I
So
see.
yeah, we're trying to generalize more, not only for metabolomics and proteomics, like more for different omics. So it used to be an MSN set one, but now everything is a summarized experiment. Sorry to
I
repeat.
see. Okay, no, thank you, Paul. Thank you for pointing that. Although here, when I run it, the reason I said MSN set is it comes up as an MSN set. Are you saying that the more recent version of POMA uses summarized experiment?
Exactly.
I see. Okay, so
The
for
first version, version of the package was based everything on MSN set or maybe Q features and all of this. But now the last version is based on summarized experiment.
I see.
Everything.
So for the purposes of this talk, then I'll just um, use the helper functions, sort of the accessor functions that are associated with this object, but just for people watching to know that it's now been now it's stored as a summarized experiment object, the newest version. Is that correct?
Yeah, I think so. Yeah.
Okay, so so we have this object here, and one way to look to see what values are associated within this object, we can use this function called exprs that comes from the biobase package. And if we just put in the name, the name of this data object, so 336, we can see in our console we see all of these the metabolites that were measured in the study and the intensities associated with the different samples. So they're either DMD or I believe healthy. And so you can see that there are lots of these samples. Paul mentioned the that these data sets contain missing values. And Missing values are tricky to deal with because you either, I don't know, you just get rid of them, which is not recommended because you're losing information, or you then have to deal with the missing values in some way. So that's called data imputation. And the great thing about POMA is it offers many ways of doing this imputation. So that's what we'll take a look at now. Um, I mentioned that this data set has some missing values. So how do you see how many missing values you have? So one thing you can do is you can use this function called is.na. And as the name suggests, it just asks a simple question, is the value na or not? And it will give you a true or false depending on the answer. So you can apply is.na not to the st00336 object, but to the output of this exprs, right? So if we run that, what we get is a bunch of mostly falses because this data set mostly contains non-missing value, but you can see that there are a few trues here and there, meaning that those correspond to missing values. So how do you see how many? That's easy. Once you have this, you can just put all of this within the sum function, right? And you have 60, so there are 61 missing values in total. So we now move on to the next part of the vignette. So Pol's written a nice description of this data set and where the data were collected from. And so now we can move on to the POMA parts. So the first function I mentioned, or Pol mentioned, was POMA impute. So if we just go to line 118 and we do run it, so question mark POMA impute, you can see this really nice help section that describes the various parameters that this function contains. And importantly, so if we look at the method here, we can see that there are different ways of doing this data imputation. You can do median, mean, Paul mentioned KNN, so KNN stands for K nearest neighbors. So that's another machine learning, algorithm. And that's something I think that hopefully in a future CBRG meetup we'll go into in more detail. So that's the one that is used in this meetup, in this vignette. And if we run that, we create a new object, and we can run POMA impute on this st000336 object, and we can save the output as an object called imputed. And if we run imputed, we see again that it is this object here. And we can just confirm that there are no missing values. So to do that, I'm just going to copy paste the code I wrote previously, put it on line 123, and then I just replace this st000336 with imputed, right? And we see that there are no missing values anymore, which is great. Now, one thing to know, and this is obviously not specific to POMA, it's just true of processing data, is sometimes things can happen to your data. So you could maybe lose a feature, lose a column, or you could lose, in this case, like a metabolite or a whole sample. And it's important to kind of keep track of your data set as you are moving along. So what I mean by that is, if we look at the dimensions of the original object, so if we do dim exprs st000336, we see that it has 31 rows and 57 columns. If we do the same thing for the imputed object, we see it also has 57 columns, but it has 30 rows. So we've gotten rid of one metabolite. And the reason for that is, if we go back to the POMA impute, it has this cutoff equals 20 parameter. And so if we look at the help section, we see cutoff numeric that indicates the percentage of missing values allowed in each group. So unfortunately, one of the metabolites had greater than 20% missing values. Is that correct, Paul?
Yeah, absolutely.
Okay. So, so kind of is like homework. I put a question here on line 127, which metabolite had, quote, too many missing values. So that's just for people at home to work out. Okay, so now moving on to the fun stuff of visualizing our data. So Paul later on, so in this case, Pol is normalizing the data. And we'll see why we like that's a desirable thing to do. So to normalize the data, it's super simple. We can use this function called POMA norm. And if I just do question mark POMA norm in the console, again, you see a really nice help section associated with this function. And it shows you that, once again, there are many options for normalizing your data. So if we look at the method section, we see that there are all of these advanced ways of normalizing your data. Now, this can be a little bit overwhelming just because there are pros and cons associated with different normalization approaches. It really does depend on what you want to do downstream. I did find, and it's also has to do with the kind of data that you are analyzing. So for metabolomics data, I found this really nice link here. So on 146, that so this specifically slide 8 of this these lecture slides on metabolomics statistics evaluates and compares these different normalization approaches. So I think this is really useful to have as a reference. Having said that, let's take a look at the effect of normalization. And to do that, one easy way is to look at the distribution of your data across the samples before and after normalization. So I'm going to do it the old-fashioned hard way. So we do exprs, imputed, and then we pipe that into, so since I want to use dplyr and ggplot, I'm going to convert it to a tibble. And actually, let's just take a look at the data as it is now. So we pipe it into view, we have this kind of thing where we have the intensities for the various metabolites for each of the different samples. Now, I want to create a box plot for each of the samples, and I want to have all the box plots. So a box plot is a useful way of visualizing distribution. I want to have box plots for all the samples. Now, unfortunately, if we go back to this data set, or this data frame, is not in the shape that I need to make these box plots. So to do that, so this is what's known as a, quote, wide data frame. I need to turn it into a long data frame. And I think it would be useful, again, to maybe dedicate a whole CBRG session to the concept of data frame reshaping. This is more of a, I don't know, I feel like it's more of a feeling thing for me. Like when I look at a data frame and I think of, oh, I want to make a certain plot, it just, the wide format just doesn't feel right. So I'll turn it into a long format and I get a really nice, I get the plot I want to get. Sorry, that's a terrible way of describing this, but at least that's how it works for me. So let's convert the wide to a long so that we can see what this, what the effect of making this data frame into a long data frame is. So we can use this function called pivot underscore longer, and I want to reshape my whole data frame, so all the columns. So I first specify the columns equals everything. Oops, not everything. And the way pivot longer works is you need to specify. So what we're doing here is we're taking all of our columns from the wide data frame and producing a new data frame that will only have two columns. One column will contain the names of all the previous columns, and the second column will contain all the corresponding values. So I need to specify a names2 parameter. So I do names2 equals, I can call that sample, and a values2 parameter, I can call that intensity. And now, if I pipe this into a view function, I see I went from a wide data set, a data frame, to now a long data frame. So I only have a samples column with all the samples and the corresponding intensities. So now, this is ready to plot. So if I do ggplot AES, and I want to have my samples on the x-axis, and I want to have my intensity on the y-axis. And since this is a box plot, I use geom box plot. Let's take a look at this as it is right now. And so we see we get, you know, something kind of okay. It's not great because you can see like all the samples, they look really, they're all bunched up here. So one way to fix that is to use a theme function. I really think it would be useful to have another CBRG session just dedicated to theme because it's such a rich function. So we can do axis.text.x is equal to element text angle equals maybe 45. And if we run that, we see that now, again, not great. I'd have to do some more work, but you can see that the labels look a little bit more legible. Anyway, so you can see that there's a little bit of work to be done here. And there's a reason I'm showing this to you. It's to compare what you would have to do by yourself to what POMA can do for you. So we have these box plots here. This is for the unnormalized data. So we run POMA norm, line 147, create a new object called normalized. And what we can do is we can again make box plots for normalized by copy pasting the previous code and just replace imputed with normalized. If we run this, we see that clearly that there's been a strong effect on the distribution across the samples, right? If we compare the previous one to this one, and again, is this a good thing? Is this a useful thing? It unfortunately really does depend on your application. And hopefully this reference here, 161, will help you decide what normalization to use. So I wrote all of this code to make this crappy plot here. Let's see what POMA can do. So if we go to line 174, we have POMA box plots. And we can apply this function to the imputed values. So these are the non-normalized values. We run that, we get this beautiful plot. And you can see that it's colored according to the sample. And whereas the previous one wasn't, the text is much more legible. It's got a nice background. It has a label, nice labels. And similarly, if you want to look at the effect after normalization, again, you do POMA box plots on the normalized object, and you get this. So much nicer, much easier. So compare line 174 to all of this code I had to write here to make an inferior plot. Alternatively, if you don't like box plots or you want to see, if you want an alternative way to visualize the distribution, if we go to line 187, this is something that we've covered previously, the so-called density plots. So there's a function called POMA density. We run that on the non-normalized values, we get this. So everything is crunched up here. And if we run it on the normalized values, we see this kind of normalization, literally normalization of the data across the samples. So this makes it very clear what the specific normalization approach we used is doing. Paul mentioned outlier detection. And I really don't know how to do this myself in R, so it's great that we have this function called POMA outliers. So we go to line 204, we run that. We see, actually, Paul, can you help me interpret this plot?
Yeah, I'm having some problem because I have some delay watching your code, and then so I'm not sure what plot you mean.
It's POMA
Which one?
outliers.
Yeah, which one? The polygon plot or the
Polygon.
box plot?
Polygon.
Yeah, these are this plot is tried to group by like samples by different groups in the call data,
Mm-hmm.
and then compute the distance from each sample to each group centroid. So we are computing the centroid of each group and then compute this distance to each group centroid. And then we can, with this distance, we can plot this polygon plot, or we can plot the outliers, the box plot, and then see if we have some samples super far from our group centroid. So we consider those samples as outliers. So
Excellent.
we cannot consider outliers from all groups at once, because we can have two different groups in our data, and this is great. So we are computing the outliers within each group.
I see. Okay, thanks, Paul. Yeah, okay, I would have had no idea how to do this by myself. This is great. And also, you used the term centroid. I think it'll be really useful to have, again, a future CBRG session where we discuss what a centroid is and how to compute distance and things like that. Thanks, Paul.
Hmm.
And now we run this function called POMA outliers and take a look at preprocessed. So POMA outliers has now removed the outliers as it says here. Okay, great. So we now have an object called preprocessed where we've normalized the data, we've imputed, normalized the data, and removed outliers, all with just 3 lines of code. So this is great. And then to wrap up, we just have 5 minutes. I'm just going to touch on a little bit of the statistical analysis. And so if you just wanted to do a regular t-test, if we go to line 220, we can use this function called POMA univariate, give it the object name preprocessed, and specify that the method is t-test. And if we run that, we get the p-values and the adjusted p-values for these t-tests comparing the controls in DMD. Everyone loves volcano plots, but there's a little bit of work to be done to code a volcano plot. So if we go to line 227, we can apply the POMA volcano plot function, and we get this nice volcano plot that shows us up and down regulated samples along with their p-values. And just to finish up, we have POMA lemma. And lemma is something that we've introduced a couple of times now. Again, all you have to do is specify the name of the object, the contrast. So the contrast in this case, you specify a contrast to specify which groups you want to compare. So which groups you want to use to compute your fold change. So contrast here, DMD minus controls. Adjust is equal to FDR. That's the false discovery rate. And you can see we get the log fold changes in the console, log fold changes, t-statistics, p-values, adjusted p-values, just as you do in lemma. And I've put this here in the interest of time. I won't write the code, but on line 238, I've written old-fashioned way. So since we've already taken a look at lemma a few times, this is a little challenge if you want to try to do it yourself. And then once again, compare that with how easy it is to do it with POMA. With that, um, so Paul, thank you again for going through this. This is super useful. Now I can avoid having to write multiple lines of code. I can just use POMA to do all of my statistical analysis. And just to finish up, you mentioned like kind of the initial motivation for making this package was to analyze specifically metabolomics and proteomics data, but you want to make it now a kind of pan-omics approach. Could you just touch on that?
Yeah, so the aim at the very beginning, was because actually POMA is not some things. Maybe this package has some new things, but we are not implementing here new statistical methods or new statistical complicated stuff. So we are just trying to make this process very much more easy. So this was the aim at the beginning, and at the same time it was for metabolomics, because I was doing my thesis, my PhD in metabolomics, so it was the first. But actually we realized that all different omics, so it's the statistical analysis can be very very close. So I know we have different, something different can be different from preprocessing. For example, normalization is not the same for metabolomics or for genomics, or so there are some steps that are, let's say, specific for each kind of omics. But actually t-test, lemma, the statistical analysis can be pretty similar, pretty close. So yeah, I decided to move from metabolomics to something more, let's say, more general.
Great. Have you considered writing a book, like a book down for POMA
Oh,
along with the theory?
oh my God, this is a lot of work.
That's true.
Maybe, I don't know. Or maybe, as you say, for another CBRG session.
Right. Yeah, it could be a collective effort.
Yeah,
Great. Thanks, Paul, and thanks to everyone for watching. And
thanks.
we'll see you. We're doing this now. We're reducing the frequency of these meetups to once every 2 weeks. So we'll see you in 2 weeks' time. And we are going to begin single cell RNA-seq analysis then. So hopefully see as many of you there as possible. Bye.
Thank you.